Hey guys, because of my discovery of DNA and how the structure is, you're able to watch TV shows like this one. Well, before we get started, please note the disclaimer listed above because in this video, we're going to be covering college biology or AP Biology's section 6.1, unit 6, section 1, which is all about the differences between DNA and RNA structure.
DNA and RNA are a very unique topic. In fact, with DNA and RNA, it is understood that one of the biggest things with biology is the central dogma. The central dogma is our story that basically says that DNA turns to RNA turns to protein, which is another fancy way of saying that guess what? Our DNA is our story that gets translated into our proteins, but not that simple. It usually goes DNA to RNA to protein, which tells us how we have different stories for each of us individually that tell us what kind of hair color we have or what kind of eye color we have, whether it be green or brown. And then what happens is we end up passing on those traits onto our offspring or our children essentially. And we utilize different processes like DNA replication, transcription, and translation to work together. And sometimes you can have mutations to create our unique identity, which is so interesting because eukaryotes have their own way of formulating or where the DNA is, in fact. DNA in eukaryotes is oftentimes in the nucleus itself and is copied during cellular division, which you might remember learning if you haven't done so when you talked about mitosis and meiosis in Unit 4, cell communication cell cycle. Or maybe you might have talked about this in Unit 2 when it talks about the cells. Prokaryotes, by the way, are DNA contained in a very unique way. So with prokaryotes itself, they have DNA that doesn't really have a nucleus. Remember, there's no membrane-bound organelles. You might have covered this in Unit 2. And it's important to note that with prokaryotes, their DNA is just a singular chromosome, and it might have plasmids, and sometimes it contains genes. Whatever it may be, we know that the DNA is less evolved than eukaryotic DNA. And the process for eukaryotes to replicate is often far more complex. This is just a brief review of maybe other units, and something that you need to understand when you're covering AP biology or college biology is the fact that when you're talking about biology itself, cells are not just a monotone topic. You're not covering it in just Unit 2 or Unit 3 or Unit 4. You're covering it as a whole. And this topic of DNA and RNA that we're going to cover really builds upon this idea that we have learned all this material and we're going to cover the nitty gritty of how genes will eventually express themselves and eventually regulate themselves in the future, which will be covered in later sections of Unit 6.
Hi, my name is deoxyribonucleic acid, and my friend over here is called RNA or ribonucleic acid. We share some similarities, but we also share some differences as well. So make sure to save this brief overview table because it's going to tell us what's the difference between both of us. This is a great overview, not just for this unit, but other units where DNA and RNA are spoken about as well. For example, we have the same sugar phosphate, but we have some differences in nitrogenous bases. For example, myself, my one unique nitrogenous base is called thymine, while my friend over here has uracil, which we're going to cover in just a second. For now, it's also helpful to understand that I'm considered double-stranded. DNA is double-stranded, while my friend over here, RNA, is considered single-stranded, meaning we're only on one side.
Let's take a look at the structure of DNA and compare it to RNA. Firstly, you need to understand that DNA means double and RNA means single. And what we mean by that is you can see here for DNA, you have two sugar phosphates, while for RNA, you have one sugar phosphate. So the comparison between DNA and RNA, we could say, is that they both have sugar phosphate backbones, but they have different amounts. So DNA has two, RNA has one.
Now let's take a look at the nitrogenous bases. So for nitrogenous bases, you have 4 letters for DNA and RNA, except the letters are a tad bit different. So for DNA, you have A, and then for RNA, you also have A. You have G for both DNA and RNA. You both have C for DNA and RNA. But the difference is thymine and uracil. So T and U. So for DNA, you have the lettering T, and for RNA, you have the lettering U. So that's the way that you can tell the difference between the two. And the letters basically represent different words of different types of nucleotides or nucleobases, you want to say. So the nitrogenous bases, I should say that, is adenine, thymine, guanine, and cytosine for DNA. And then for RNA, it's adenine, uracil, guanine, and cytosine. So for thymine, it's for DNA, and for uracil, it's RNA. So you could see that they both have 4 nitrogenous bases. However, the lettering is considered different when you compare thymine to uracil. And I want to stress this because this difference can come up in multiple choice questions, and it can often be cumbersome if you don't recognize the differences between the two. Now, going back to the bigger picture here, you can see that there are nucleobases or nitrogenous bases that basically line up RNA on the one side. Yet for DNA, you could see that they pair up with each other. So we call them base pairs because basically you have these nitrogenous bases that can connect to one another. So what you need to remember is the connections are always the same for DNA. So you always will have the same connections for DNA as such. So you'll have A that attaches to T and C that attaches to G. So that's the difference between the two. And then for RNA, you want to replace the T if it is binding to an RNA structure to a U. So what you need to understand is that for RNA, it is single-stranded, but when you get into processes like transcription and translation, which you'll cover in a later unit, you need to understand that DNA can attach to RNA and eventually will become protein. And because of this, you'll have like a pairing of a DNA that attaches to an RNA. So it's understandable to see that the T will be replaced with U. You don't need to know that for right now. Just be aware that RNA is single-stranded and that the pair that is once thymine or T for DNA is now replaced with uracil or U. And that's helpful to remember, especially when we're discovering processes that are related to the central dogma, which we'll cover later on, which is DNA going to RNA, going to protein. And because of this, you could see that RNA can have its own attachments. It's also safe to remember and understand that A attaches to T for DNA and C attaches to G. Now, with these nitrogenous bases that attach to each other, it's helpful to remember that the A attached to the T has 2 hydrogen bonds that connect them together. So you can see here that there's this 1 bond and 2 bond. And then G attaching to C, so G attaching to C over here, will have 3 hydrogen bonds, 1, 2, 3. I don't know if you've taken chemistry before, but you want to remember this little tidbit fact for you. The more bonds that you have between the bases, the stronger they are. So essentially the G to C connection will be stronger than the A to T connection, which means that the A to T connection will be easier to break. A little fun fact for you. And that's because there's 2 hydrogenous bases, or 2 hydrogen bonds, I should say, that connect the bonds together between A and T. And then G and C have those 3 strong bonds that are together. Now, remember, there's thymine with the DNA, and you want to remember that behind the thymine is what we call the deoxyribose for DNA. Now, with the nitrogenous bases for RNA, you'll see that the backbone is actually ribose. So that's the difference between the backbone structures. Like the backbone, the sugar backbone that's affiliated with the attachment for DNA is going to be deoxyribose, and for RNA is going to be ribose. And then they both have a phosphate attachment, which is in the backbone that we talk about, the sugar phosphate. So to recap, you have the sugar phosphate that is in both DNA and RNA. However, for DNA, you have two strands, and RNA, you have one strand. The base pairs are attached for DNA to one another, where you have A attached to T, which has two hydrogen bonds, while G attaching to C has three hydrogen bonds. And then you also want to remember that RNA can attach to other bases when it comes to processes that are affiliated with the central dogma, which basically states that DNA can turn to RNA, which can turn to protein, which you'll learn in a later unit, which means that A that once was attached to T will now be replaced with the letter U, or uracil, if you're talking about RNA. So the nitrogenous bases for RNA are very similar with G, A, C when it comes to the similarities between DNA and RNA. The difference between RNA and DNA when it comes to nitrogenous bases is that thymine, it will be replaced with uracil, or U. It's also to remember that you have two different backbones. For deoxyribose, you have a deoxyribose sugar, while RNA will have a ribose sugar. They both have the same phosphates that are attaching, which means that they have the same sugar phosphates. So this covers the structure and the similarities and differences between DNA and RNA. Okay, I want to highlight something really important, and it's the fact that when we're talking about the nitrogenous bases or the nucleotides, we're talking about the fact that DNA has A attaching to T. However, for RNA, A will attach to U instead of T. So when we talk about the other bases, so C and G, for both DNA and RNA, they will always bind together. So you will always see cytosine binding with guanine for both DNA and RNA. This is really critical to remember, because remember, we talked about the fact that DNA is double-stranded while RNA is single-stranded. Why is this important for you? Because you might receive a free response question, wink, wink, or you could receive a multiple choice question, wink, wink, that will ask you to calculate the total percentage of nucleotides that exist in a chain. And you need to understand that's on a 100% scale. However, you'll see that A will bind to T always if it's DNA, or A will bind to U if it's RNA. And those percentages will be exactly the same. So for example, if adenine is 20%, guess what? That means that uracil will also be 20%, which means that the total will be 40%, right? Because 20 plus 20 is 40, which means that if you subtract it from 100, that 40%, that will give you 60% left over to compensate for the cytosine attaching to guanine, which means essentially if you take 60 divided by 2, that gives you 30% each for cytosine and guanine. So that will essentially create this structure of either DNA or an RNA that is complete at 100%. So I just wanted to give you this higher level conceptual thinking when you think about scenarios that you potentially could see. Don't worry, we'll might cover this in a later problem down the line. But I just want you to remember for now that DNA, as you can see here, is double-stranded and RNA is single-stranded. And while you might be thinking, when am I going to base pair the RNA? Remember the central dogma that you may or may not have covered in a previous unit when you're talking about transcription and translation. Remember, DNA goes to RNA, goes to protein. And during that transition between DNA and RNA to protein, there is some pairing involved for RNA. So just keep that in mind as well when you conceptually think about the differences between DNA and RNA. Let's talk about the term purines, and let's talk about the term pyrimidines. So when we're talking about the nucleotides, we categorize them into two main groups. To recap, remember we said that A usually attaches to T and C usually attaches to G. And if we're talking about RNA, remember that T would be replaced with a U. So adenine is considered a purine, and guanine is also considered a purine. They also have, remember, cytosine, which we consider a pyrimidine, uracil, which is considered a pyrimidine, and thymine, which is considered also a pyrimidine. Some things to note that when you have the attachments, you have one purine attached to one pyrimidine. So, for example, A or adenine, which is a purine, attaches to T or U, which is considered a pyrimidine. So one purine will always attach to one pyrimidine in terms of forming the bond. Now, the other thing to note is I'm going to clear my screen here, is the reason why we categorize purines and pyrimidines together is not because we want you to memorize more things than you need, but because of their structures themselves. For example, when we take a look at adenine and guanine, remember, they're never going to bind to each other. Binding adenine to guanine is never going to happen. You have one purine attached to one pyrimidine, but adenine and guanine have very similar structures in the sense that you have double rings. So you see there's a ring on this structure and another ring on the same structure. So there's 2 rings. Same thing with guanine. We have one ring here and one ring here. 1, 2. So purines considered to have 2 structures of rings, so double ring structure we call them. In difference, we have pyrimidines, and pyrimidines are single ring structures. So you can see it's one ring here, and one ring here, and one ring here. So essentially what happens is you have one purine which has a double structure, which will attach to one pyrimidine that has one solo structure, ring structure. So basically you have one purine with one pyrimidine. And the purpose of this is to ensure stability. If you remember, we have the double ring and the single string that we want to ensure they stay that way, they don't break apart. And the purpose of having this idea of having these bonds attached to each other with one purine and one pyrimidine, it sort of balances each other out and creates a sense of stability and allows it to last in the appropriate time frame necessary for success. Let's take a look at this hypothetical quiz question and see if we can answer the question together. Before you get started, if you feel confident in answering the question, feel free to pause the video now as we're planning to go over how to solve this problem together. Okay, now that you're done reviewing this problem and potentially getting the correct answer, let's see if you got the correct answer. So firstly, the correct answer is C, and let me tell you how we got there. So it talks about a double-stranded DNA, and they tell you that there's 540 total nucleotide subunits. So total subunits and base pairings let you know that it's going to be 540 divided by 2 is going to give you 270 base pairs. I'm going to use BP as an abbreviation to represent base pairs. So that means that you're expecting that 27 or 270 base pairs exist. Okay, so that means that A attaching to T and C attaching to G. This would be representative of one base pair, and this would be representative of another base pair. So we know in total, each one of these nucleotides, remember, is 540. So I just want to point that out as well so you can see the relationship of what we're talking about here. They want to know the percentage of subunits of guanine. So they're letting you know that there is also exactly 81 subunits of adenine. And remember, you will need to know that A is attached to T and C is attached to G. Therefore, if we have 81 of adenine, that also means that we have 81 of thymine. So they're going to be together, right? Because those are subunits and they match up or attach to each other. So in total, we have the AT combo, which is 81 plus 81, which ends up giving you 162 subunits. Because we have 180 or 162 subunits, I'm looking at what we have is guanine, which is the leftover, right? So remember, in total, we have 540 subunits, right? So we can say, I'm going to use a different color here. We have 540 subunits minus our 162 subunits because of the AT combo is going to end up giving you the C. I'm going to make that a G or C or CG or GC, either one combination. So when I go ahead and try to solve for that, I end up getting 378 subunits. So that must mean that this is guanine and cytosine combined, right? Because C attaches to G. If I go ahead and divide by 2, remember, I'm going to attain what I'm looking for, which is the guanine, right? So I'm going to take that 378 and divide by 2 because that's going to be half of the pair because remember, one side is G and the other side is C. And if I take the whole set and divide it in half, I get the one side that I need, which is the guanine. So when I go ahead and do that, I get 189. So essentially, I have 189 guanines that I need. But remember, they're asking for the percentage. So percentage is part over total, and that you should know from math, right? So just to recuperate that percentage, I'm going to say 189 divided by part over total, which is 540, because remember, I had 540 total and I had 189 for my part. And then I'm going to times that by 100. And the reason why it's 100 is because it's converting to percent or 2 decimal places to the right. So when I take 189 divided by 540 and times it by 100, I get 35% or 0.35 with 2 decimal places to the right. And so the correct answer is 35. I hope this question helps solve some of your problems that you might see for the idea of AP biology related questions. Make sure to hit that subscribe button because we're updating our videos for the new year, and we hope that you stick around so you could see these questions and more like this in the future. Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our link tree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams.